Yeah, now you are the host, Narendra.
Yeah, fine, sir. Thank you. Anyways, so very good morning to all of you, and a very special thanks to Professor Suresh Babu as well as Vishwajit sir for giving me an opportunity to address you again. In fact, Suresh Babu sir being my mentor, my teacher, he is giving repeated chance to me to address Bangalore University students. I don't know whether this is because he goes on giving chances until I give a good lecture. Anyways, so in fact, I have divided the lecture into two parts. Today I'm going to give you the part one, and the second part will be on Saturday. So in this part, rather, the title I've bit modified. Now it's peptide conformations. Actually, the tentative title was the Ramachandran Plot, these principles and applications. That I will reserve for my next class, because today I'm not going to address anything on Ramachandran Plot. Rather, I just want you to brief about the peptide conformations, and at that background, how the Ramachandran Plot helps us. So, if I have to start my lecture, you know, the stereochemistry, peptide conformations or any conformations are based on the same principles of stereochemistry, what we study in our MSc, the conformations and configurations. So if very popular question any stereochemistry teacher begins with, what's the difference between a configuration and a conformation? So if any of the student can answer, it will be a bit encouraging to me to go ahead. Configuration and conformation. Can I expect anyone to answer?
No, you can't.
Why? It's okay.
They know the answer, so they don't want to tell you.
They know. They don't want to share their knowledge with me. Okay,
yeah,
fine.
yeah.
It's still okay, sir. No problem. I mean, this is the typical student. Even our students are like this, sir. They
Yeah.
answers to none of the questions. Anyways, so the configurations are the spatial arrangement. Both configuration and conformations are relative spatial arrangements of atoms and groups. Both are same in spatial arrangement, but in configuration, the spatial arrangement is locked, fixed due to the bonding itself. But in conformation, the spatial arrangement is not fixed. It is very flexible. It is not locked by chemical bonding or simply covalent bonding. It is not locked by covalent bonding. So if you have to change the configuration of a molecule, the only way to do that is you have to break the bond and reform it in the opposite direction. That's the way. So for example, you can take L-lactic acid, L-alanine, or you can take D sugars. You can't make them its enantiomers unless you conduct a chemical reaction, breaking the bond and doing it from the opposite direction. But conformations are not like that. Conformations, of course, they are spatial arrangement of atoms, relative spatial arrangements. But as I told you, they are very flexible. You can change a conformation of a molecule simply by at least you can fill up this blank. You can change a conformation of the molecule by what? Okay, by simple rotations of the essentially the rotations of the single bonds. Although double and triple bonds are not free to rotate. So you conformations. Okay, this is what conformations are. So when we talk about conformations, that comes into picture immediately with molecules as simple as ethane. Is it not? Ethane has two carbon atoms connected by a CC bond. So now you can rotate the CC bond, right? Because it's a single bond. Now, as you know, you would have studied the basics of conformations, that ethane can exist in, which are the two conformations ethane exists in? Of course, this is not two important conformations or two extreme conformations which we know, very popular conformations of ethane. Eclipsed and staggered, is it not? So in the eclipsed conformations, all the hydrogens are almost touching each other, whereas in the staggered conformations, hydrogens are as apart as possible. So if I ask you a question, how many conformations ethane can exist? What would be your answer? How many conformations ethane can exist? Okay, so yeah, somebody was giving the answer.
6.
6 conformations. So which are those? Of course, this is not an interview. Yeah, very feel free to interact. Which are those? Of course, 2 I have already named. 3. Usually the ready answer we get for this question is 3 conformations. Which are those? Eclipsed, staggered, and skewed. Is it not? But but uh, uh, but I'm not telling this is a wrong answer given by students, but this is only a partially correct answer. If correctly we have to answer this question, how many conformations ethane exist? Innumerable. Is it not? Innumerable. Why? Because yes. So you are sending your answers in chat, no problem, fine. Why? Because how do you distinguish different conformations? We use a term called dihedral angle. So with eclipsed conformation, we tell dihedral angle is 0. With staggered conformation, we tell dihedral angle is 180 degrees. But there is no restriction on the molecule to have dihedral angle anywhere in between 0 and 180. It is still possible, right? You can have infinite conformations. You can have innumerable conformations. But why are we so obsessed with eclipsed and staggered? Because they present extremes of their energies. Is it not? So of all the given conformations possible, of the infinite conformations I am telling you, eclipsed is the highest energetic, whereas staggered is lowest energetic. There are innumerable conformations of intermediate energy in between. That's fine. So academically, these 2 conformations interest us. So what is that I'm stressing to tell you is a simple molecule like ethane, just 2 carbon atoms, 6 hydrogen atoms, has innumerable conformations. And then you look at the propane. Look at propane. Of course, it has many conformations, out of which we again take staggered conformation to be the least energetic conformations. Why is this energy and conformations go hand in hand? Why? Because molecules try to occupy a conformation or try to possess a conformation which is least energetic. Is it not? So if you take some 1,000 molecules of ethane, and if you are having a technology to see individual molecule, which are the conformations that exist, you can see some 99% of those molecules will be in staggered conformations. Why? Because that's the lowest energy conformation. So all molecules try to occupy the lowest energy conformations always. Of course, you can change the conformations by simply supplying heat, giving more energy to the bonds, they rotate, and then more population in the eclipsed higher energy conformations also. Anyways, at ordinary temperatures, it's the lowest energy conformations. That's the lowest energy conformation of, this is the lowest energy conformation of propane. Okay. You see the lowest energy conformation of butane. Of all the possible conformations, this is the lowest energy. And you know, we use usually Newman's projection to depict conformations. Newman's projection of butane. You can see the anti-periplanar or anti-parallel arrangement of 2 methyl groups. By the way, why does an eclipsed conformation become unstable? Why does an eclipsed conformation become unstable? Can I expect an answer? Why does eclipse, not just with ethane, with any molecule, why are eclipsed conformations unstable? Yeah, you can send it. If you answer directly, it's okay. Due to steric entrance. Very good. Somebody gave the answers due to steric entrance. That's the answer basically. So what happens in the eclipsed conformations? the atoms, see, now the carbon here and the carbon, they are bonded carbons. So these bonded groups or atoms always have an attraction. There's no problem with this. But what is the type of interaction with non-bonded atoms? Example, the hydrogen at the back carbon here and the hydrogen in the front carbon. What is the type of interaction between these two? Are they attractive in nature? No, because they are not bonded. Bonded atoms are attractive in nature, whereas non-bonded atoms are repulsive in nature. Non-bonded atoms are simply like gaseous atoms. They cannot come too close. There is collision and they start to move apart. You know, maybe that some simple kinetic theory of gases. So two atoms cannot come too much closer, not beyond their van der Waals radii, right? They cannot come so much closer. So these two atoms cannot come very close. But if you force the molecule to happen like that, how do you force it? By rotating this bond. How do you rotate it? By giving temperature. So these two start repelling. So the conformation becomes high energetic. And with small atoms like H, if there is so much repulsion, what about bulky groups like CH3 and CH3? Can you ever imagine, can you imagine even attempting bringing these two groups very close in the eclipsed conformation? No, almost not possible. It is possible, very high energy conformations are possible at very high temperatures, but generally they are not possible at all. Okay. Why am I telling this? Because the same principles hold good for peptide conformations also. Okay. Now, last one. Hexane. What is the conformation of the hexane? You can see the puckered nature of the hexane, right? The zigzag conformations. Carbon down, up, down, up, down, up, down, up. Why a zigzag conformation is adopted for a hydrocarbon? Why a zigzag conformation is adopted for a hydrocarbon? Basically, when we write the structures also on paper or on board, when we write hydrocarbons chain, we write like this in a zigzag wavy manner. Why is that? Why is that? Because this zigzag conformation is the least energy conformation. This is the conformation which represent the point or the conformation where all atoms are at maximum distance. So in a way, the repulsion between the non-bonded atoms is at its minimum. Therefore, zigzag conformations are the one which is. Okay, fine. So this is what a quick recap on the conformations principles. Okay. Now I'll ask you a question. Let us construct a peptide example with 6 carbon atoms or 6 backbone atoms. Right? Or maybe 10, for example. Even a decane exists in zigzag conformation. So if I construct a peptide with 10 backbone carbon atoms, of course, in between there will be nitrogen atoms also in the backbone of the peptide bond. Anyway, let us take an hypothetical example. I have constructed a peptide with 10 backbone atoms. Now, with analogy to decane, what should be its conformation? What should be its conformation? With analogy to decane. What?
Staggered.
Staggered. I mean, staggered zigzag. Simply.
Zigzag.
Zigzag. But if you look at the conformations of peptides, that is not always right. There are peptides which exist in the zigzag conformations. There are peptides which deviate from the zigzag conformation. In fact, to our surprise, we see that even small peptides can exist in very compact helix-like, ring-like conformations. So what is this? Why is this happening then? Is it not? Why is it deviating the general laws of conformations? Why is it not minimizing its energy by adopting a zigzag conformations? Okay, so these are the questions which we are going to try to answer in our in today's and in the next class. Why? What is the driving force behind peptide conformations? So when you extend this analogy, when we extend this argument from peptides, which are small molecules, to proteins, then what happens? Anyways, so let us not. So to proteins, what happens? It so happens that a protein, there is something called a new term coined for protein conformations called protein foldings. Fine. So when we call about protein foldings, that again is a conformation term. Folding is also a conformation term, but an extreme state of conformation, foldings. So these are the things happen. And what we are going to try to is find some basic answers for such questions. Why are protein turns? Why are peptide bands? Why are peptide forms helix? These are the things we are going to address in our next questions. Anyways, so I hope this torsional angle you are familiar with. This term torsional angle we are familiar with. Why the torsional angle concept is required? Because as I am going to tell you in my later slides, these are the torsional angles are the indices of pinpointing a conformation. Okay, see now, for example, if I ask you to tell conformation of staggered ethane without writing its Newman's projection or structure, can you still tell it? Describe it in one line verbally. Can you describe? We can still describe, right? What can I tell? The dihedral angle of that conformation is 180 degrees. If I tell this much, it is more than enough. So using that, I can write a structure. So torsional angles are the index of conformations. Although you can tell, I can write a structure and show how the conformation is, that is still fine. But that happens when your molecules are smaller, ethane, propane. But when the molecules becomes larger, then you cannot write the structures. You cannot write the structures over every carbon-carbon bond, or you cannot write the conformations over every carbon-carbon bond. But at least you can tell what is the torsional angle at this carbon C2, C3. What is the torsional angle between C5, C6? Such things you can write. Okay. So torsional angles are the index of conformations. So what are these torsional angles? Again, a quick recap. I know that you may be familiar, but sitting at home for almost 3-4 months, I myself have forgotten a lot of subject. Expecting students to know everything is not fair from our side. Anyways, so what is torsional angle? It's almost like your dihedral angle, except for one term. See, yeah, I'll just give you this difference. In dihedral angle, you will never find a negative side in a dihedral angle. Even if you read volumes on conformations. Example, you open standard stereochemistry books like Nasipuri or Kalsi. You see dihedral angles, never you get negative dihedral angle, but torsion angle you get negative sides. Okay, so what are these torsional angles? Torsional angles are the angle between, I'm just want to bring out some important point here. Usually the way we have conceived this term angle is what? The angle between two lines, right? Which are joined by a point, like the angle inside a triangle. That's what we have conceived. But torsional angles are different. These torsional angles are not angles between adjoining lines. Please remember. Instead, it's the angle between two lines which are further apart. They are not adjoining. Like see, AB is the line here, CD is the line here. Now the torsional angle is between AB and CD, and AB and CD are not joined. Okay, so in a way, torsional angles are some spatial parameters, some distant, faraway term parameters. So this is what a torsional angle is. At least you should have 4 atoms to make a torsional angle. At least 4 atoms. A, B, C, D are our 4 atoms, and then you have to hold one bond towards you. Like in this picture, I have held AB bond towards me, and look at how your DC bond is oriented with respect to AB bond. So you can do like this, like BC is free to rotate. So what is the possible torsional angle? 360 degrees possible, right? So what is the distance between them? If you take this, you see this again, the concept with the simple lines, I've extended it to a molecule. You can see a molecule like this. Here is a group. Any 4 atoms I'm going to choose. You can choose any 4 atoms, A, B, C, D. And these 4 atoms, don't choose from the same carbon atom, like 1, 2, 3, 4. You don't choose these 3 atoms of the methyl groups and make a torsional angle. 4 atoms from the neighboring carbon atoms or neighboring atoms, general, because it's organic molecule. I'm just telling carbon atoms, neighboring carbon atoms, A, B, C, D. And this is your viewpoint. Look at AB. And how is CD oriented? Maybe this is just some 30 degree, right? Plus 30 degree will be your torsional angle between A and D. Please remember, torsional angle between A and D is 30 degree. So you can go on rotate like this until you get D in the parallel position, almost opposite direction of A. Then the torsional angle will be plus 80 degree. And further rotations, then that becomes anti-clockwise. You can measure from here. For example, if my, okay, I don't have one more slide. If my D is here, okay, if my D is here, what is the torsional angle? D is here. What is the torsional angle? This is the position of D. What's the torsional angle? Yeah, I'm getting answers in that. 90 degree. Yes, correct. 90 degree. Fine. There is no problem. But what is the sign of the torsional angle? Is it plus 90 or minus 90? This is my question. Because I told you, torsional angles have sign, unlike dihedral angles. Minus 90. Very good. Yeah. So I can see answers coming from you. Correct answers coming from you. So if D is here, it is plus 90. If D is here, it is minus 90. Why? Because now you see how your arrow mark is there from A to D. This is in the clockwise. Is it not? But if you have an arrow mark like this, it is anti-clockwise. Therefore, this is minus 90 if D is here, whereas if D is here, it is plus 90. So I hope that you are now familiar with this important concept, very simple concept of torsion angle. Although it appears simple, but it is a very significant concept when we try to analyze peptide conformations. Okay, so please remember at least one thing from my lecture is about these torsional angles, how to imagine them, and how to get the positive and negative signs of the torsional angles. Okay, fine. Now, yes, now let us move on to conformations of peptides. So till now we were discussing conformations of simple molecules, maybe some 4, 6 carbon atom, 10 carbon atom molecules. Now let us come to the conformations of peptides. As you know, peptide is a macromolecule, not as bigger as a protein, but relatively a big molecule. Unlike your organic compounds, which are smaller in size, peptides is a large molecule. Now, if you are interested in the conformations of peptides, you should, if you ask the question, a given peptide which I have synthesized or which I have isolated from nature, what would be its conformations? See, I'm not asking a question like this. What would be its structure? Then this is not a very complete question to ask. Why? I mean, because structure is a very general term. Please remember, conformations and configurations are also part of structure. So we can call that structure as an hierarchy. So if we looking at a very bare minimum bonding of any molecule, I'm not just telling peptides, bonding, which atom is connected to which atom, which group is connected to which atom. Such simple questions you are asking wherever, then that becomes a primary structure. Okay, so if you write the structure of ethane as CH3, then what is that structure? It's a primary structure. Okay, but the world doesn't end at this primary structure level. In fact, if you look at molecules, primary structure, of course, it's there, primary structure, but many of the properties of the molecules are the functions of conformations and configurations. So primary structure is not that important for us anyways. But when you look at the conformations of ethanes, it is not primary structure. Then you are going to one more level up in the structure hierarchy. So you're looking at conformations. Similarly, a peptide conformation. So that's what I'm not asking what is the structure of the peptide, because structure of a peptide is a very general question. You can just tell, you can give any answer to this question. What is the structure of the peptide? Structure of the peptide is this tripeptide is made up of alanine, glycine, alanine. Simple. But I'm asking what is the conformation of the peptide? Then we have to be a bit careful in answering this question. Fine. Now, proteins or peptides. Although protein conformation is too much for us to discuss, all my lecture will be pitched only at peptide level. But these are the some terms which we use interchangeably. If I call a protein conformations or a peptide conformation, usually we use interchangeably, so no problem. So how do we represent the conformation of a peptide? You know, strictly speaking, a conformation has to be analyzed across any two bonds which are free to rotate. Is it not? A conformation has to be analyzed across any two bonds, or any bond which is free to rotate. So if there are some 200 carbon-carbon bonds in a molecule, at every carbon-carbon bond, you have to analyze the conformations. If I rotate throughout 360, what are the different spatial positions of atoms? But that we don't do in proteins and peptides because of enormous task, that is enormous task. And sometimes such an enormous task doesn't become so much satisfying and meaningful at the last. So rather we have simplified this a bit. You know the basic architecture of a peptide, right? Okay, nice. Let me go to the peptide structure. I don't have a peptide. Anyways, you know the basic architecture of a peptide. So what is a peptide? Basic architecture of a peptide. You have amino acids which are joined by the dehydration reaction to form a copolymer of amino acids. So what is a copolymer? Copolymer is a one which is formed by different monomeric residues. You can have peptides with same monomers also, like a polyalanine peptide. You can have only alanine throughout the peptide. But usually peptides come in diversity. So you have the copolymer. And what is the structure of a typical structure of an amino acid? You have alpha carbon, carboxylic group attached to it, amino group attached to it, hydrogen atom attached to it. The rest will be your side chain. Depending on side chain, you have different amino acids. But when we are doing structural conformational analysis of a protein, we are simply ignoring the side chains. Okay. So how are side chains placed? We are not worried. What is that we are worried? How is the backbone placed? So in the sense, what do I mean? What is the conformation of the backbone? So this complex picture of a peptide or a protein can be simplified by omitting, at least pictorially omitting the side chains. So your peptide chain boils down to its backbone only. Now, how is this backbone arranged? What is the conformation of the backbone? This is the question. Like you can take a thread, like I'm just giving an example from our daily life. You can just take a thread and you can extend it, you can fold it, bend it, make coils out of it, make a pellet out of it. You can do anything you want with a thread. So imagine this peptide backbone as a thread. And what is the conformation of this thread? Whether it is coiled, whether it is extended, whether it is forming a loops, knots, all these things. Same thing here. So I just have to remove side chains and see the backbone structure only. That is the peptide conformations or proteins conformations. Okay. Somebody can still argue it is very wrong because why are you ignoring side chains in the conformations? But the answer is we don't do it. We only take the backbone conformations. Okay. So yes. So coming back to our first question. If I take a peptide like its hydrocarbon analog, does it always exist in zigzag conformation? No. The answer is no. Instead, it comes in different conformations. In fact, If you look at all these conformations and helix, beta sheet, or a turn, which I'm going to show later, they are all very pleasing to eyes. They are all a delight to watch, to look at the pictures of conformations. Like, the double helix model of DNA. You may look into some, what is that, throwback pics of Watson and Crick holding a ball and stick model of DNA. If you Google it, you get, it's very good, nice to look at. The model itself is so attractive. So the peptide structures are very pleasing to eyes. Fine. So this is an helix, what I have got. I mean, you please keep track of the backbone atoms only now. So which are these backbone atoms? CN, CC, N. C, CC, N, CC, like that. It's just the backbone atoms. What are these backbone atoms? They are nothing but the amino acids which are formed the peptide bonds. And these are the side chains, R, 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 R. How is your backbone? If you start from one end to the other end, it coils. The backbone has coiled like this. So in a way, it is like your spiral staircase. Fine. I hope at least some of you would have seen spiral staircases in some buildings. Spiral staircase. So the railing of this staircase, you just follow the railing of this staircase. What happens? From one end of the railing to the other end of the railing, it doesn't come in a straight line, straight down, or just goes straight up. Rather, it bends on its own axis, comes back. You can look it up from a top view also. There's a side view of the helix which I have shown you. You can also start from this view. Look from the top. How does it look like? Like a snail's shell. So what I want to tell from this slide is that helices are not uncommon. Whether it is a peptide or a man-made world like a staircase or a natural world like a snail, helices do exist. Coils do exist. Anyways, these two are different reasons for being coiled. These are different reasons for being coiled. Anyways, so I just wanted to give this diagram so the impressive images we see, the better for your remembering at least. So these are the helices. You can also have peptides in beta sheets. So what is a beta sheet? Now comes your zigzag conformations. Zigzag extended conformations of peptide chain held together by hydrogen bonds. These are beta sheets. You can also have a bend, turn. This is called turn. You have, see, the chain is going like this, turned, and then come back. So this is one more conformation. So these are the three important conformations of a peptide: helix, and then beta sheets, and then turns. Of course, there are classifications within helix: 310, 312, such helices, right-handed helix, left-handed helix. Even beta sheet, there are parallel beta sheets, anti-parallel beta sheets, and turns can be alpha turn, beta turn. There are classifications within these fundamental structures. Anyways, so if you account for all these classifications and put together, what are all the possible structures of peptides? You get lot of possibilities. Lot of possibilities a peptide can exist in. So now we are going to reason out why it is existing in a particular structure. Okay, fine. Just a quick recap about amino acid chemistry. Amino acid chemistry, you have alpha carbon. I think I can skip these types. These are the side chains of amino acids, and you see, anyways, not very much connected to my topic. A general inquiry. There are many polymers in nature other than peptides, proteins also. Starch itself is a polymer, right? Glycogen is a polymer. And then you have nucleic acids, one more class of polymers. But in none of the polymers, there is so much diversity in its structure, like in peptides and proteins. In starch, what is it? The repeating unit is simply glucose, entire glucose. And in DNA and RNA, the repeating units are only 4, A, G, C, T. But in peptides and proteins, the repeating units are 20. And even with AGCT, where is the difference in the nitrogenous base? Very minute difference. You have purines and pyrimidines, single cycle and bicycle, nitrogenous heterocycles. But you look at the side chains of all these 20 amino acids, lot of diversity is there. Smallest H, glycine, smallest side chain being glycine, to very big side chains like the one with like the one in histidine, like the one in phenylalanine, phenyl ring, or a proline ring structure. There is a lot of diversity in the side chains of amino acids. So why this was required by nature? That's the answer again, because of the diverse functions of proteins. Anyways, not addressed. Now, yes, now we will quickly go into the relationship between the primary structure and the secondary structure. Like, what did I tell? The primary structure of a protein or a peptide is simply nothing but the amino acid sequence. So from N terminus to C terminus, if you go, which are the amino acids you encounter? That is the primary structure. So simply primary structure, you can just write down alanine, glycine, phenylalanine, go on writing down like that. The sequence of amino acid is the primary structure. Now, if we analyze the database of all the peptides whose secondary structures have been solved, I mean, this is a big task by itself, is it not? You know, big data analysis, all these are the terms which are used in computers now, in information technology. Almost bioinformatics and proteomics, genomics, these are all the domains of big data analysis, of course. Anyway, so if you make this big data analysis and try to get some meaning out of this analysis, we can form a meaning like this. What is that meaning? In the helices, these are the amino acids which are mostly present: alanine, cysteine, leucine, methionine, etc., etc. In the beta sheets, these are the amino acids which are present. In the turns, the bends and the loops which I showed you, these are the amino acids which are present. So we can just make one interconnection here. What is that? By and large, I am not telling exclusively, by and large, the secondary structure of the protein is predetermined by the primary structure, by and large. Of course, there are exceptions, by and large. Okay, fine. So you can take alanine, cysteine, leucine and form a helix out of it. You can sit valine, isoleucine, phenylalanine and form a beta sheet out of it. Make a peptide containing only these amino acids. The most probable structure would be a beta sheet. If you take only alanine, cysteine, leucine of the first group and make a polypeptide out of it, most probable structure will be an helix. Fine. Okay. So I know now we will start our actual analysis, conformational analysis of the peptide bond. So if you look at the peptide bond, how is it? You have the amide bond, CONH. Either sides of this, you have an alpha carbon atom. Please remember, you have an amide or a peptide bond, which is called, it is not amide. You know, Suresh Babu sir will definitely grow angry if I call this an amide bond. It's a peptide bond. So on either sides of this peptide bond, there are alpha carbon atoms. And alpha carbon atoms, of course, has its own side chains. But like, you know, I told you one thing, right? What is the thing I told? Strictly speaking, you should do this torsional angles determination across every bond. So can I do a torsional? These are our three fundamental bonds for a conformational analysis of a peptide bond. Which are those three fundamental bonds? Bond between carbonyl of the peptide bond and C alpha. Bond between carbonyl of the peptide bond and the nitrogen of the peptide bond. Bond between nitrogen of the peptide bond and the next C alpha, next amino acid C alpha. So you should do analysis. You should turn these bond and see what are the conformations possible. You should turn this bond, these bonds and see what are the conformations possible. But luckily enough, or fortunately, at least to make our analysis easier, understanding easier, this CN bond is not free to rotate. CN bond of the peptide bond is not free to rotate. Why? Because you know, amide bond because of resonance, you have a partial double bond character in an amide bond. So one bond we have fixed. We need not do a torsional angles possibilities of CN bond of a peptide bond, fortunately. To that extent, our analysis has been simplified. Okay. So which are the two remaining, which are the two, okay. So which are the two bonds for which we have to do the torsional angle analysis? Carbonyl of the peptide bond C alpha and the nitrogen of the peptide bond C alpha. There is one more way of looking into this. Instead of placing our what? I'll just go back. Instead of placing our peptide bond at the center of our analysis, we will put a C alpha at the center of our analysis. So what are there on the either sides of C alpha? On one side, the C alpha is connected to carbonyl carbon of the peptide bond. On the other side, the C alpha is connected to nitrogen of the peptide bond. And this bond is free to rotate. Of course, free to rotate. C alpha. NC alpha is also free to rotate. Which is not free to rotate? This bond. Peptide bond is not free to rotate. And this peptide bond is not free to rotate. So if it is not free to rotate, it is a double bond character. What is the geometry of a double bond? What is the geometry of a double bond? And question. What is the geometry of a double bond? What's the structure? Geometry. We shouldn't call a structure because structure is most for a molecule. For a bonding, it is geometry. What is the geometry? Is it tetrahedral, trigonal planar, something like that? Answer, anybody giving? What's the geometry of a double bond? Yeah, I have received an answer. Planar, good. The double bond is planar. That's the plane I have shown here. This is the plane I have shown here. So when it is planar, the adjacent, the 6 atoms are in one plane. 1, 2, 3, 4, 5, 6. Similarly, 1, 2, 3, 4, 5, 6. So if you are rotating this bond, yes, there is one more implication of this. Very important implication. If you rotate this bond, you are rotating the whole plane with it. Yeah, trigonal planar, geometry of a double bond. That is okay. Trigonal planar, good. Planar, basically planar. You are actually rotating the plane. Although if you rotate this CC bond, the whole plane rotates. So 1, 2, 3, 4. 1, 2, 3, 4. 4 atoms rotate with this, along with this bond when you rotate this. Similarly, when you rotate this bond, C alpha N bond, these 1, 2, 3, 4 atoms rotate with it. Okay, fine. So this is one more picture of, anyways, let us define torsional angles here. Now we have two torsional angles. How are they defined? The torsional angle due to the rotations of C alpha C. That is called psi. The torsional angle originating from rotation of C alpha N. That is referred to as phi. Okay. So a simplified picture of that I have just showed here. C alpha N phi, C alpha C psi. These are the two torsional angles. Okay. So like I said, again, one more better picture of what I have been telling, phi and psi, phi and psi. So essentially what is the point? That you rotate bonds which are now freely rotatable, C alpha N and C alpha C. Okay, that's fine. Now, like I said before, you can describe a conformation by specifying its torsional angles values. Like for staggered conformation of ethane, it is 180 degree. For eclipse, it is 0 degree. Similarly, now, is it possible to us to define the conformation of this part of the peptide? Please remember, this part of the peptide, not the entire peptide, not the entire peptide, this part of the peptide by giving the values of phi and psi. It is possible, right? Okay, So like this, you just theoretically 360-degree rotation is possible here. Theoretically 360-degree rotation is possible here. Okay. So for every conformation, you just rotate this and write the phi value, psi value. There is one more way of doing this. You fix a value of psi. For example, psi you fix at 90 degree. What are the allowed values? What are the possible values of phi? You can go on doing like this. And this, if you repeat around every C alpha in a peptide, I have just put a part of the peptide and done this torsional angle values, given the torsional angle values. There is one more C alpha here. There is one more C alpha here. So this C alpha C will be our psi. C alpha N will be our phi. See here, C alpha N will be our phi. C alpha C of the next amino acid, which is not in the picture, will be our psi. So go on doing like this for every amino acid in the peptide. Take C alpha as your central point. Find out the values of phi and psi. Find out the values of phi and psi. For what? For an amino acid, for the peptide. And list it out. List it out. What are the phi values at amino acid number 1? However, anyways, I'll just tell you one thing here. If you have 100 amino acids, the terminal amino acids are not analyzed conformationally. So you should have 98 sets of value. For the second amino acid, third, go on till 99th amino acid. For every amino acid, write what is the, for every C alpha, write psi value, phi value, psi value, phi value. And then put it in the form of a graph. You will see that you are essentially, you have, I'm just hinting towards my next lecture. Essentially, you would have constructed a Ramachandran plot. Okay. So Ramachandran plot is that you have, okay. I think I should stop it. I just draw a picture of how the rotations take place. It's a picture of how the rotations take place. Fine. Yes. So your phi rotates, psi rotates. They take different angles. Okay. Fine. Now, let me stop at this point because I see I have exhausted 55 minutes. Next time we have a class. So simply we'll ask this question very directly. Are all phi? Oh my God. Yeah. Okay. So is any value of psi and phi possible for an amino acid residue inside a peptide? Okay. Yes. So if we'll ask this question, is any value of phi and psi possible for an amino acid residue inside a peptide? Okay. If your answer is yes, any value is possible at the given temperature, say at room temperature, any value is possible. If this is our answer, or if this is our discovery, scientific discovery, then the conformational analysis of peptides become meaningless. Is it not? Then you just can give one line answer. A given peptide can exist in any possible conformations. Innumerable conformations. Very, very flexible, highly flexible. But actually, what is the scientific discovery? On the contrary, any value of phi and psi is not possible. For every amino acid in a peptide, only some values of phi and psi are possible. Very narrow range of values of phi and psi are possible. So why any value is not allowed? Because of restrictions to the rotations. Please remember here, this restriction to the rotation is not coming from a double bond. Rather, it is coming from steric hindrances. Steric hindrances of which I'm going to show you later in next steric hindrances of the amino acid residue side chain along with peptide bonds carbonyl and peptide bonds nitrogen H. These are the steric hindrances which don't allow phi and psi to take any values. Okay, so that is what we have to do in our next class. What are the values of psi and psi? Possible values of phi and psi in nature for peptides, for natural peptides. What are the possibly allowed values of 5 and 7? Okay. So anyways, yes. So now comes our question. At this question, I will just leave you. Next class, we are going to answer this question. What is the question? Is it possible to predict the most stable conformation of a peptide when its primary sequence of amino acid is moved? Okay, this is my question. I hope there are hints to the answer in my presentation itself, like there are some amino acids which are helix makers. There are some amino acids which are beta sheet makers. There are some amino acids which are turn makers. That is a gross answer. Is it possible to find prediction of the structure when the amino acid sequence is known? Okay. So if we do a prediction of this, that becomes the application of Ramachandran plot. So Ramachandran plot is helpful for us to predicting the conformation of a peptide when its amino acid sequence is given. Okay. So this I will reserve for my next class. Okay. Fine.